So we're going to be comparing instant devices with laboratory testing. The instant devices come in all shapes and forms. There are dips, and there are cups, and there are cassettes. There are dips that have multiple analytes on each channel. They come in various shapes. Circular. Here's one that has a multiple number of analytes on each channel. Instant test devices have been around for a very long time. In fact, one of the earliest instant test devices that was around showed up in the late 70s, early 80s, and these were not for drug testing. Drug testing devices are fairly recent, but this was for pregnancy tests, initially used in physicians' offices and laboratories, and later on went into the home testing market. So instant devices in all shapes and forms for various different drugs and hormones and compounds have been for a long, long time. And as I said, they're very, very popular. Now, opposed to the instant devices that are laboratory testing. Laboratory testing is performed on large automated clinical chemistry analyzers that can perform testing very, very efficiently. Reagent cost is relatively low. The throughput on some of these devices, on some of these instruments, you know, they can push through hundreds and hundreds of samples on an hourly basis. So it's basically a comparison between laboratory testing and instant devices. Now, the instant drug testing devices come under a variety of names, and I may use these interchangeably. I tend to be kind of sloppy in this regard, but some places are called field test kits, instant drug test, on-site drug test cups, drug test dips, drug test cassettes, home drug test. Many of the pharmacies, the drugstore sell home drug testing for use in the home. Another name is point of care drug testing. Basically, regardless of the name, the devices are essentially the same. Now, there are two types of instant test devices. There are those devices that are CLIA waived, and basically the manufacturer sends supporting documentation to the FDA indicating that these devices are very simple to perform the testing, and basically if you get CLIA waiver, then they're non-regulated. Virtually anyone with training in the use of these devices can perform the test. Same thing goes with the home devices, home test devices. They're relatively simple. Now there's another category of test device, and these are FDA cleared, and these are classified as moderate complexity testing. These are primarily used in a laboratory setting or in a office setting, and there are FDA requirements for the type of personnel who are allowed to do moderate complexity testing. As a comparison, screening on the large automated clinical chemistry analyzers are also classified as moderate complexity testing. If we look at the menu of tests that are available on the instant devices, depending upon which manufacturer you get the instant devices, the menu is fairly large. I've listed some number of analytes here. Some manufacturers may have other drugs that they can test for, but most of the manufacturers have testing for the basic group of drugs. These include amphetamine, methamphetamine. These may be a separate channel for AMP and meth, or it may be a combined channel barbiturates, benzodiazepines, buprenorphine, the cannabinoids, and by this I mean the natural cannabinoids, testing for use of marijuana, cocaine or cocaine metabolite, MDMA, or ecstasy, alcohol, ethanol. There are some kits out there for ethylglucuronide, fentanyl, methadone, and methadone metabolite. There are devices out there for opiates. These are primarily looking for morphine, codeine, hydrocodone, and hydromorphone, depending upon how the device is configured. Then there are separate channels for oxycodone, PCP, phencyclidine, propoxyphene. Then there are devices for synthetic cannabinoids. These are for the spice, the K2 type of compounds, tramadol, tricyclic antidepressants, TCA. And some manufacturers have channels on their devices for specimen validity testing. Some devices may include creatinine, specific gravity, pH, and oxidants. And of course, the menu varies tremendously from one manufacturer to the other. For specimen validity testing for creatinine, the results are essentially qualitative, giving you an idea of where the creatinine level lies. You do not get a true quantitative result for that. So how do these devices work? They actually use a fairly ingenious type of chemistry. All of the point of care devices, as well as the tests that we use in the laboratory, use antibodies to recognize the drugs of interest. So antibodies are generated against the drugs or the family of drugs that we're interested in, and there is some sort of a colored signal that's used as an endpoint. On the point of care devices, that endpoint is visual. For the laboratory-based testing, the endpoint is actually read spectrophotometrically on the instruments that we use. So let's just look briefly at how the instant devices work and compare that with how the laboratory tests are formed, or the mechanism for using that. Virtually all of the instant devices use some variant of lateral flow immunoassay. So here is a diagram of how the typical lateral flow assay works. There's essentially some sort of a surface membrane onto which some sort of porous material is placed on that allows the specimen to wick up the sample. If you look at the sample here, there's various areas on the test strips. There's a sample pad. This is where either the specimen is, a couple of drops of specimen are put on the sample pad, or if it's a dip, this is inserted into the urine container. Then there's a conjugate pad. The conjugate pad includes antibodies to antibodies that are specific to the particular drug. And these antibodies are labeled with some sort of a label that when an immune complex is formed, that label has a colored endpoint. Colloidal gold is often used, and that imparts a reddish-brown color. So the sample is put on the pad, or the dipstick device is inserted into the specimen. The specimen wicks up, picks up the conjugate from the conjugate pad, the reaction membrane where everything is mixed up. Then the sample wicks up further up the test strip. And if you look at the bottom panel here, these antibodies are directed against the drug that's of interest. If there is drug present in the particular sample, the drug binds to the antibody that's embedded onto in the test line area. And I'll show an example of that a little bit. If drug is bound to the antibody, then the marker antibody cannot bind to the antibodies that are generated against the drug. Then in the control line, these are the same antibodies to the drug, but the conjugate is directed against the drug antibody, and this is basically a control line. So the conjugate antibody binds to these antibodies, the immune complex is formed, and a color is generated. And I'll show an example of that in a little bit. If we look in the right-hand panel, this sort of shows a schematic of how the chemistry on the large automated clinical chemistry analyzers work. We use a technique called enzyme multiplied immunoassay technique. It's abbreviated EMIT. This is a proprietary method. This method has been around since the mid to late 1970s. There are a number of variants of this. Different manufacturers use a variant of it. But I'll describe basically how it works in a very simple schematic form. Again, there are antibodies that are in one of the reagents. These antibodies are directed against the drug of interest. This is the drug of interest. There's also the drug of interest that is labeled with an enzyme. This enzyme can act upon a substrate, and when it acts upon the substrate, another compound is formed that we can measure spectrophotometrically. So into the sample is introduced antibody and enzyme labeled drug. You incubate it, and there's a competition for binding sites on the antibody between the drug in the specimen and the labeled drug. Now, there's a unique characteristic of the method that when the labeled drug is bound to the antibody, the enzyme is inactive. That means that it cannot act upon the substrate. So we set up this competing reaction, and the amount of enzyme labeled drug that's available depends upon how much drug there is in the patient's or the donor's specimen. We go through a number of manipulations. We add substrate to this as enzyme can act on the substrate. That forms a product, and that product we can measure spectrophotometrically, and then the computer and the instrument can calculate the concentration and tell us if the specimen has drug or not. Okay, the next slide shows a schematic of the instant test devices. This is the way they look at it. They've got two areas, an area called C, which is a control, and an area called T, which is a test. Now, this is a very simple test strip. It only measures one drug. It could be any drug. There are devices that have multiple channels that will have multiple test sections. Each test section is specific for a drug metabolite, or a family of drugs. So this end of the test strip is introduced into the sample. Either sample is placed on it, or if it's a dipstick, it's put into the urine specimen. The specimen wicks up. The urine sample wicks up. It picks up the conjugate. And if there's no drug in the specimen, then there's a colored reaction of both the test area and the control area. Reason we get a color in the test area is there's no drug there to compete with binding sites for those enzymes. So those binding sites on that antibody are not blocked. So the conjugate binds to those sites, and it forms a colored complex. Now, if there is drug in the specimen, the drug in the urine sample occupies all of the binding sites to the antibodies in the test section. The conjugate then cannot bind to them, and there is no color generated. So a positive result occurs when, or the result is called positive when there is absolutely no color in the test area of the device. The control area must light up. It must produce a color in order for the test to be considered valid. You can think of the control strip as a built-in quality control or quality assurance measurement. If the control area doesn't display color, then something has gone wrong in that test. One of the things that could occur is the donor might have added some kind of an adulterant to the urine sample, which would invalidate the test. So here's an example of a instant device that is invalid. There is color in the test zone, but there is no color in the control area. So this test would be considered an invalid test specimen, would necessitate new collection and a new test on this particular specimen. So in a nutshell, this is basically how it works. So if there's color in both the control and the test area, then that specimen is called negative. If the test area is completely devoid of color, and this sometimes becomes a little bit subjective, but the control area has to be, the color has to be absolutely devoid from this area to call a specimen positive. So that does result in some subjectivity on the part of the tester. And with the built-in control line, if for some reason the control line doesn't display color, then the test is considered invalid, and either negative or positive results cannot be reported from that specimen. Okay, the next couple of slides look at the results of a published study and some studies that were performed internally at Cordant, looking at how the various devices work. The first slide is a summary slide from a study that was published in Clinical Chemistry in 2014, about 2.5 years ago. The study was performed in the Department of Laboratory Medicine at the University of Washington Medical School in Seattle, Washington. The senior author is Dr. Hofnagel, who happens to be on the faculty in the Department of Laboratory Medicine. What they did in this study is they looked at 1,681 urine samples. All of these samples had confirmatory testing performed by LC-MS, liquid chromatography, tandem mass spec. So this is a confirmation method. This is a well-accepted gold standard or platinum standard for confirmatory testing, if you will. All 1,600 plus specimens were also analyzed on a CLIA wave device, an instant test device, and they compared the results in 5 different lots of this particular CLIA wave device. So if we look at the chart here, we've got results from 5 different lots of the device. They were looking at 3 particular groups of analytes. They were looking at opiates. This is a test that is calibrated against morphine. It's very good at picking up morphine as well as codeine, hydrocodone, hydromorphone. Does not pick up oxycodone or oxymorphone terribly well. The second group of drugs that they were looking at was oxycodone. This particular test device recognizes both oxycodone and its primary metabolite, oxymorphone. On the right-hand side of the panel, they were looking at methadone, which is commonly used in methadone treatment programs. So they were looking at both the false positive and the false positive rate as compared to the confirmatory LC-MS-MS method. So let's put our attention first on opiates, and let's look at the false positive rate for opiates. All 5 lots of this particular device had false positive rates for opiates less than 5%. Now that's, in my mind, that's pretty good. Pretty good results. They ranged anywhere from just under 5% in lot 1 to, this is approximately 1.5% false positive rate. I think this is lot 4. So, you know, less than 5% false positive rate is actually pretty good for opiates. They also looked at the false negative rate. These are specimens that on the on-site devices tested negative, but on the confirmatory test, there was positive opiate that was listed there. So the false negative rate for opiates ranged from approximately, this is approximately 2% in lot 2, up to a high as over a little bit over 12% in lot 5. So for the false negative rate for opiates, there's quite a bit of variability from lot 1 through lot 5, through the different lots that they were looking at. So from a very low false negative rate to a relatively high false negative rate, 12% is, in my opinion, is quite high. Next, they looked at oxycodone, the false positive rate. The false positive rate for the oxycodone on the instant devices ranged from about 11–12% up to a high of just a tad over 25%. Quite a bit of variability from lot to lot, from 12% approximately to a little over 25%. And so if you're using lot 2, approximately 1 out of 4 specimens that tested positive on this instant device for oxycodone was a false positive result. fairly high. Now let's look at false negative rates for the oxycodone test, and they vary from a low of less than 1% false negative rate, which is excellent performance, to a false negative rate about 15%, which in my opinion is high, but quite a bit of variability from lot to lot, from less than 1% to a high of approximately 15%. Now, if you compare the false positive and false negative rates for the oxycodone, the high false positive rate was not correlated with a high false negative rate. So high false positive rate, but a high false negative rate. So exactly what's going on with this lot, I do not know, but quite a bit of variability. For methadone, the false positive rate for most of the lots were fairly consistent at a little bit under 5%, but lot 5 had a false positive rate that was approaching 20%. The false negative rate across all 5 lots was fairly respectable, right around the 1.5 to 2% false negative rate, except for lot 2 that had a false negative rate of approximately 5%. So the take-home message from this published study is that depending upon which analyte you're looking at, there could be a lot of variation from lot to lot from the same manufacturer for both false negative and false positive rates. And depending upon what analyte it is that you're looking at or which drug class you're looking at, the false positive rate can be very, very high, and the false negative rate, for the oxycodone and here for this lot of methadone, can be fairly high. I'm sorry, this false negative for opiates and false negative for oxycodone can be quite high and can vary quite a bit between lot to lot of the same manufacturer's agents. Okay, the next couple of slides are data from an in-house study. This study was performed at our Long Island Cordant Health Solutions laboratory, directed by Dr. Damon Porg. And what they were looking at, they looked at 400 urine specimens that were subjected to both confirmatory testing by liquid chromatography and mass spectrometry, and each sample was also tested on a point of care device. There were approximately 4,500 different tests that were performed on the 400 samples. So multiple tests were performed on the same sample. So if we look at the first slide from this study, this is looking at the false positive rate according to drug class distribution. And there's quite a bit of variability here. For the amphetamine class, the false positive rate was about 9.5%. For barbiturates, about 44%. False positive rate for buprenorphine, Suboxone was essentially 0%. Benzodiazepines, 23%. Coke performed very well. The cocaine, benzoylecgonine for methamphetamine, about 50%. Opiates, 22%. Methadone, 16%. Oxycodone, about 13%. PCP is 100%, but there was only one specimen that was false positive, so we can't attribute too much to that. Tricyclics performed fairly well. THC, about 22%. If we look at the bottom portion of this slide, this actually shows the actual number of specimens that had false positive. Remember, the total number of specimens was over 400. Out of the 400 specimens, 21 had positive point of care devices. 2 turned out to be false positives, so a false positive rate about 9.5%. The significant ones I think we need to look at are methadone. There were 63 positives, 10 were false positives, about 16% false positive rate. Opiates, about 214 positive results, 48 were false positives, false positive rate about 22%. Oxycodone, 205 positives, 26 were false positives. So false positive rate about 12%. As I mentioned a little bit earlier, the false positive rate for PCV was 100%, but we only had one sample that tested positive, and it was a false positive. We really can't attribute too much to that. There were no positive tricyclics, so obviously the false positive rate is 0. For THC, marijuana metabolite, we had 9 positive results. 2 of them were false positives, so about a 22% false positive rate. Now, since the study was performed at a laboratory that primarily deals with medical monitoring for pain clinics, they don't see a lot of some of the drugs that we would be seeing, say, in a laboratory that primarily looks at criminal justice or treatment. So you have to look at the data in that light in terms of the population that they're testing. The second slide from this series of studies is looking at the false negative rate for the various drug classifications. So for the amphetamines, it's about 4%, buprenorphine about 3%. Again, these are false negatives where the on-site device gave a negative result, and according to the LC-MS confirmatory method, they were able to detect drug in these specimens. The benzodiazepine rate false negative rate is fairly high at 15%. Coke is very good. Methamphetamine, we missed about 1.5%. They missed about 1.5%. For opiates, approximately 11%. Oxycodone, about 6%. Now, if you look at the table down below, this actually looks at the number of actual specimens that tested negative and the false negative rate. So remember, again, there were 400 specimens. For amphetamines, there were about 380 negative results. 4% were false negatives. Again, the comparison method being definitive confirmatory methods. For barbiturates, very, very low false negative rate. That's good. For buprenorphine, very low false negative rate. For benzodiazepines, the false negative rate was about 15%. This is actually quite comparable to the laboratory testing, where benzodiazepines, when you're looking at a very, very complex family of drugs, both the false positive and the false negative rate can be very high. One of the things with the benzodiazepine screening method, it does not pick up clonazepam, or its primary metabolite 7-aminoclonazepam, very well. So a number of these false negatives results is 49 may be due to the presence of clonazepam. Cocaine, there were no false negative results. Methamphetamine 6 for about 1.5%. Skip down to opiates, there's about 20 false negatives on the opiate, 12 for corresponding false negative rate, about 10%, 11%, 6%. There were no false negatives for phencyclidine, tricyclics, or THC. This next slide is sort of a summary looking at the incorrect point of care results by drug class. And if we look at this graph, we're looking at both the false negative rate in blue, the false positive rate in green. And basically what this graph tells us is those percentage of samples that were tested by the point of care device that did not agree with the results obtained from LC-MS. And a couple of things are striking here. The incorrect rate for benzodiazepines, opiate, and oxycodone are very high, in my opinion. Moderate rates for amphetamine, methamphetamine, and methadone. Now, if you recall from the study that was published in Clinical Chemistry, the results that came from the University of Washington Department of Laboratory Medicine, they were looking at opiates, they were looking at oxycodone, and they were looking at methadone. And in those three three groups of drugs, they had a fairly high rate of false negatives and false positives. And this is what we're seeing. This is what my colleagues at Long Island saw, a fairly high incorrect rate for opiates, oxycodone, methadone, and benzodiazepines. Now, the study out of clinical chemistry, they did not look at benzodiazepines at all, so we have no comparison data there. The next slide is sort of a summary slide of the study that was produced at Cordant Health Solutions in our Long Island facility in Huntington, New York. The study was performed under the leadership of Dr. Damon Ward, my counterpart in Long Island. And as a reminder, they looked at 400 urine specimens, and a total of 4,500 tests were performed on those 400 specimens. So approximately 11 tests were performed on each of these specimens. So if we look at the upper left on this chart, this is looking at the positive point of care devices. This is looking at all of the drug classifications. So the false positive rate in this entire group was about 18%. The true positivity rate was about 81%. This is a little bit higher than we would expect from false positive rates, comparing them to laboratory testing. The negative PO's point of care results, about 97% of the negative results obtained from the point of care testing were indeed true negatives. Overall, there was about a 3% false negative rate across all drug classifications using the point of care device. Now, if we look at the final pie chart here, this is looking at basically the correlation or the concordance of the LC-MS results, which we're saying is the gold standard, the definitive results, and the results that we obtained from the point of care device. These numbers may surprise you a little bit, but just under 60% concordance between the point of care results and the LC-MS results, and about 41% of the results between the LC-MS results and the point of care results did not occur. This is basically looking at the number of individual tests, not the number of specimens that did not agree. So the number of test results did not agree. So let's sort of look, what could be the cause of some of the false positive results? And this applies to the point of care testing as well as to the laboratory-based testing. One of the reasons could be over-the-counter medications, especially decongestants for amphetamine and methamphetamine screening tests, cross-reacting drugs, may be due to other drugs that cross-react. An example would be sertraline, a very popular antidepressant, which is notorious for causing false positive benzodiazepine results. They could be drug metabolites. Not everyone metabolizes a drug in a similar fashion, so there may be accumulation of drug metabolites, especially if there's other pathophysiology involving liver or kidneys. And so drug metabolites are a potential cause for false positive results. Herbal preparations, we occasionally hear anecdotal results. Someone is using herbal preparations, results in false positive test results. When they stop using these herbal preparations, the false positive results disappear. What's in those herbal preparations that causes false positives, for the most part, is unknown. For the point of care devices, I mentioned that the absence of color is the criteria to be used for interpretation of positive results. So there could be subjective interpretation of positive test results. An individual looks at the test strip, looks at the test line, and determines that there's no color. There may be faint color there. So there's some subjective interpretation of the results. It's possible that there are heterologous antibodies. If someone has what we might consider leaky kidneys, it's possible to see heterophile heterologous antibodies in a urine sample, which causes problems with the immunoassay. And this can occur for both point-of-care testing devices as well as for the laboratory devices. Another reason might be that the cutoff levels for the point-of-care device is too low. It's too sensitive, and we do the confirmatory testing. We don't look at levels that low, potential problem. And the final reason for false positive results could be what I call nonspecific immunoreactivity. And what this is really a fancy way of saying that we really don't know what's causing those false positive results. And for most cases, we really don't know what's causing the false positive results. It's just one of the vagaries of doing screening tests by using immunologic methods. Turning attention to what might cause false negative results. One cause of false negative results may be the urine specimen is too dilute. For the immunoassay, the specimen may very well have been too dilute for the immunoassay, either the point-of-care device to pick it up, whereas the LC-MS, MS methods are much more sensitive to pick them up. Another reason for false negative results is the limit of detection might be too high in the point-of-care devices. So they're not sensitive enough to pick up the drug, which can be picked up in the confirmatory LC-MS method. So let's sort of look at the pros and cons of the instant test devices. As I mentioned earlier, they're very, very popular. They're usually very, very easy to administer. So let's look at some of the pros and cons. One of the pros is instant results, and this is very important in terms of giving instant feedback to the client. You know, you can give them, you get negative results, you can say, "Attaboy, you're doing very, very well." Or on the other hand, if they're positive results, you can immediately discuss the results with the patient or with the donor. So the pros are instant feedback in terms of the results, and the negative results, for the most part, are very, very reliable. Some of the negatives or sort of the cons of using the instant test devices is that the cutoffs may not be very, very well established. In the manufacture of these devices, it's not possible to manufacture these with very exacting cutoffs just due to the methodologies that are used. So established cutoffs may not be quite as good as they are with the testing that's performed in laboratory. One of the negatives, as we saw, is the false positive rate is very high. This, of course, varies with the drug or the drug class that is being analyzed. There's subjective interpretation of the test results. An individual looks and say, is there color, is there not color? So there's a little bit of subjectivity there, whereas on the laboratory testing, there's absolutely no subjectivity. It either is positive or is not positive. Specimenability tests are not always reliable on the point-of-care devices. For creatinine, especially, you get a qualitative result that the creatinine might be between certain levels. So to determine which samples are dilute might be difficult in some cases. And as we saw from the published study out of the University of Washington Department of Laboratory Medicine, at least in that study, the batch variability was quite high, both in terms of false negative rate and false positive rate. So this is something to consider when you're using these devices. What is the variability from lot to lot or from batch to batch of the instant devices? The variability from reagent lots on the laboratory testing is minimal, and we can control for that. It's very difficult to control for on the instant So let's compare laboratory testing with instant drug test kits. For laboratory testing, we have well-defined cutoffs, the reproducibility at the cutoff levels. Again, this is a little bit dependent upon the analyte, is probably on the order of 3 to 4%. On the instant devices is probably considerably more. Specimen validity testing and laboratory testing is quantitative. We can give you very good numbers regarding the creatinine, the pH, oxidants, or specific gravity. We have a permanent record of these results. We get a digital printout of the results from the instrument. There are well-defined quality control, quality assurance programs that can be subscribed to in laboratory testing. There is proficiency testing available. Laboratory-based testing, for the most part, is relatively inexpensive, certainly inexpensive compared to the instant test devices. One of the negatives, if you will, about laboratory testing is that the turnaround time can be long. It can be anywhere from 12 to 24 hours, depending upon how quickly the specimen arrives in the laboratory compared to when it's collected from the individual. Now, on the instant test devices, as we talked about earlier, you get instant results. The negative results are, for the most part, very, very reliable. The positive results, the false positive rate can be kind of high. The other thing with the instant devices, you can give instant feedback to your donors or to your patients. You can discuss the results, whether they're negative or presumptive positive, but at least these can be discussed. Excuse me. These can be discussed with the patient or with the donors.